hand over to Miko.
Okay.
Miko, you can start.
Paolo, did you start recording?
Yes, I've started recording.
Okay, good morning, everyone. Today is my great pleasure to introduce my colleague Alberto Giacomello as our speaker. So Alberto Giacomello is an associate professor at the Sapienza University in Rome. So he recently was successful in getting the ERC starting grant, and now he's his group. And Gonzalo Paolo, our master student, recently has joined his group, and I'm very happy about that. So Alberto also graduated from La Sapienza in mechanical engineering. Then he went for a couple of years to Max Planck Institute in Stuttgart, where I met him, and we started several joint projects. He's an expert in the atomistic and DFT simulations, and today he will tell us about the behavior of fluids and liquids at extremely small scales. Go ahead, Alberto.
Thank you very much, Miko, for the introduction and for this opportunity to talk about...
Excuse me, Alberto, excuse me. Sorry for the interruption. I just need to warn everybody that this seminar is being recorded, and by staying on, you're giving consent to your voice and image being recorded and stored. Thank you. Sorry about that, Alberto. Please go on.
No problem. So I'm very happy for this opportunity to talk about some recent work about drying in nano-confinement. I find this topic particularly intriguing because it's really like seeing water boiling at ambient condition just due to the fact that it's being confined. So it is to me a very fascinating subject, and it seems that nature is really exploiting this kind of phenomena in order to get some special properties, for instance, to plants. So this is a famous example of a lotus leaf, which is known to be always clean. And in recent years, so in the late 90s, it has been discovered that this is linked to a peculiar structure in which there are hydrophobic wax crystals on the scale of tens of nanometers, and structured surface. You see here these papillae on the scale of 20 microns. This gives rise to a gaseous pockets in between liquid droplets and the leaf itself. So this leads to superhydrophobic properties, including self-cleaning. So this has been exploited in engineering and in technology in realizing bio-inspired surfaces. So here you see some nanoscale textured superhydrophobic surfaces. We have worked quite extensively on that with Miko in recent years. Other systems in which confinement, hydrophobic confinement plays a role and induces drying are nanoporous materials. So recently, hydrophobic nanoporous materials have been proposed in several applications. In particular, the main idea is that you have liquid water all around surrounding these granules of nanoporous material, and you accumulate energy in the form of surface energy when water intrudes, so wets the pores, whereas energy is released when drying occurs, such that the pores are dry again. This seems to play a role also in chromatography, where granules of hydrophobic nanoporous material is often used as the stationary phase in the chromatography column. Today, however, I will focus a little bit more on biological confinement. And the key idea is that hydrophobic residues of proteins can be the source of confinement. So here you see an example in which there's a hydrophobic chain, and when two hydrophobic beads come sufficiently close together, they induce a localized drying, which gives rise to a strong attractive force between hydrophobic residues. This is the so-called hydrophobic effect, and plays an important role in protein folding, molecular recognition, and membrane assembly. Today I will focus on a specific instance, which I will have in mind rather than focus, a specific instance, which is that of ion channels. So ion channels are transmembrane proteins, so they go through the membrane and are, so to say, the nanoscale switches of our cells, which are able to switch on or off ionic current through the membrane. So basically they make our brain works in neuron firing or our muscle contract. So they play a rather important role for us today and always. And there has been emerging experimental and numerical evidence that many of these channels switch on or off by the formation of a bubble. So here you see two configurations of these ion channels. One in which the hydrophobic residues are further apart, in which case there's no bubble inside of the hydrophobic cavity of the pore. In this case, the ions can translocate through the pore with their hydration shell, and the gate is open. So there is ion current going through the pore, whereas if the channel is restricted, so the hydrophobic residues are closer together, then a nanoscale bubble forms, which prevents the ions to carry with them their hydration shell, such that really the gate is blocked and no current passes through. So you see that there is this mechanism of switching on and off the currents, which is really related to drying in this extreme biological confinement. So let's start our journey through drying in confinement by what we know in microscopic theory and in a very simplified setup, which is a slit pore. So imagine an infinite slit pore with a distance between the two parallel flat walls, L. In this case, you can write down the grand potential for the confined liquid phase and confined vapor phase, and this will consist in two terms. One is the volume term, which is multiplied by the thermodynamic, the intensive thermodynamic variable, which in this case is the pressure. And you have positive term, which is the cost of forming two interfaces with the wall. So this is the surface tension. And in the case of a liquid, you will have the liquid solid surface tension. In the case of a vapor, you will have the vapor solid surface tension. So when you equate the grand potential in the slit for the vapor and for the liquid, you get the coexistence conditions. So these coexistence conditions are a difference between the liquid pressure and the vapor pressure. What's interesting is that, of course, in the bulk, this would the coexistence condition would be equal to 0, and instead you immediately see that in a slit pore they depend on two main things. One is the difference of surface tension between the vapor and the confined vapor and the confined liquid, and they depend on the degree of confinement, which in this case is encoded by the distance between the wall, L. So by using Young's law, you can really relate this difference in surface tension to the contact angle. And you see that you can get positive delta P, so you can get drying in this slit pore when cosine theta is negative, which occurs for hydrophobic walls, which means that the Young contact angle should be larger than 90 degrees. The other thing that I would like to remark here is that when you go towards nanoscales, these effects are greatly magnified in that there is a dependence on which goes as 1 over L of these coexistence condition. Okay, so you can get really a stable vapor phase in rather exotic conditions as compared to the bulk, of course. the fact that you have confinement also changes dramatically the kinetics of drying. Here you have a typical nucleation scenario in which the free energy barrier arises because of the competition of favorable bulk terms. So the bulk terms tell us that a phase transition wants to occur, and instead surface terms are related to the cost of forming a bubble. So the cost of forming an interface, okay? The competition is eventually won by bulk terms which scales as the radius to the cube, and they give rise to a free energy barrier. So the time for any nucleation event is really the exponential of the free energy barrier. Now, what's interesting is that you can compute analytically the free energy barrier for nucleation in the bulk, which means a critical bubble, a spherical critical bubble, or at a flat wall, in which case is a section of a spherical bubble, which meets the surface with the correct contact angle. So in this case, you see that there's a factor psi multiplying the homogeneous free energy barrier, and this factor goes from 1 for hydrophilic substrate, perfectly hydrophilic substrate with contact angle 0, to 0 for perfectly hydrophobic substrates. So this really means that the presence of a flat hydrophobic wall, which is nothing really exceptional in the sense that it's flat, dramatically facilitates, accelerates drying, just because it allows for a smaller critical bubble to occur. Okay, so up to the fact that for extremely hydrophobic surfaces, it makes the barrier vanish. Then I also would like to call your attention on this graphical construction, which is a very classical picture dating almost 1 century ago, in which you have a flat wall, in which case the critical bubble is a section of a sphere, but then it is easy to see what is the role of the curvature of this substrate, because for concave substrates, you have that the correct contact angle is met for very small critical bubbles, whereas if you have a convex one, the critical bubble grows. This also tells you that concave, inward concavity tends to accelerate drying, whereas outward concavity does the opposite. Okay, so summing up this part, we have an important role of drying. So confinement facilitates drying by 3 main ingredients. One is the size of confinement, the other one is the surface chemistry of confinement, and the third one is the geometry, or if you wish, the curvature here. Now, the system we have in mind, ion channels today, are rather complex in this sense, because they have a rather complex geometry. Actually, it's even elastic if you want to add further complexity. They have a complex chemistry because the residues the proteins have differ going down into the channel. So there are hydrophobic, hydrophilic, charged residues, any kind of residues. And furthermore, the confinement is so extreme that one expects nanoscale effects to be relevant in this case. So how do we tackle this? Well, we resort to, of course, the thermodynamic framework, but also to resolve these, all these details to molecular dynamics simulations. The second challenge this sort of pseudo-phase transition has is that typically the 2 phases, so the confined liquid and the confined vapor, are separated by free energy barriers, which may be larger or much larger than kBt. So the microscopic timescale in which the dynamics occurs is usually much, much shorter than the timescale to observe a thermally activated crossing of the free energy barrier. So in simulation, all of you that do simulation know that the fact that you have multiple timescale, it's a bit of a pain in the neck, because you have to solve for two distinct timescales. And the other thing is that the properties at the nanoscale really influence the macroscopic properties of the system. So if you think of the energy application that I've introduced before, what happens in a single nanoscale pore determines the properties of the whole device, which is in charge of storing energy. Okay, so there's a multiplicity of time scales and length scales. And for the time scales, we resort to rare event methods. It's a specialized class of sampling methods which allows to reconstruct the thermally activated events. Okay, so this was, so to say, the appetizer for drying in confinement. And now the two main courses are, first of all, concern the stability of surface nanobubbles, and the second course will be about gas-induced drying in nanopores or ion channels. Please feel free to interrupt me with questions even during the seminar at any time, so we keep it interactive. Okay, so first course. I'd like to go for the running title, which is rather provocative and it's not boring as the actual title. Our question was, what is really nanoscale about nanobubbles? Because the main explanation of the stability of surface nanobubbles is that nanobubbles are long-lived, so they can be seen in experiments because they are generated in supersaturated conditions and because they have contact line pinning. So you see here in this IFM micrography that the contact line doesn't move as the bubble deflates. Okay, so these two ingredients, which are purely macroscopic ingredients, so there's nothing really nano about nanobubbles so far, account for the fact that nanobubbles can survive for hours and so be experimentally detected. Okay, this just because diffusion is slowed down by by supersaturation and by the fact that with pinning the curvature of the bubble decreases a lot. Okay. But this leaves some open questions. For example, nanobubbles are known to form also in undersaturated liquid. What is the mechanism here? And the other thing that this theory cannot really distinguish why they form preferentially at hydrophobic substrates rather than at a hydrophilic one. And there are other questions which are related to the nature of the liquids in which they form. So recently in 2018, an improvement of the basic model of contact line pinning and diffusion has been proposed, in which an empirical gas-solid potential has been added. And this empirical gas-solid potential solves two important issues. The first one is that it predicts a contact angle for the nanobubble, which is in line with experiment, and also the time scale of for the lifetime of the nanobubbles is correctly captured. And the second one, it accounts for the difference between attractive and repulsive interaction of the gas with the wall. Okay. However, what's still completely, what was, I hope, completely unknown is the physical origin of this potential. So what we did is try to investigate microscopically what happens for a gas at a wall and try to see whether what was the origin of this interaction potential. In order to do that, we did some simulation, restrained molecular dynamics, which is a sort of umbrella sampling of oxygen or nitrogen molecules, which of course are the main components of air, at a wall. This wall can be tuned from hydrophobic to hydrophilic, And we have a rather standard water molecule, which is TIP4P 2005. So we control the distance from the wall, and we reconstruct the free energy as a function of the distance of the center of mass from the wall. Then we take this microscopic information and we use it as an input for the microscopic model in the previous slide, which, however, in this case has been informed by first principle, so to say. Okay, so this is our main result. You see the free energy as a function of the center of mass, and what you immediately notice is that there's a minimum in the free energy which corresponds to a supersaturation in the right-hand side of the gas at the wall. This occurs very close to the wall, so to say close to the first layer in the density of water at the wall, and its depth is of the order of 5 kBt, where K is the Boltzmann constant and T is the temperature. What's interesting is that depending on the hydrophobicity of the wall with the fluid, the local supersaturation can be more than 1,000 times more than the concentration of the gas in the bulk. Okay, so there's a very strong local concentration. Then you can compare the attraction of the gas to the wall in a vacuum, which is the black symbols, with the complete system encompassing gas, wall, and liquid. And you immediately see that depending on the hydrophobicity of the wall, this enhances the presence of our liquid, enhances the attraction of the gas to the wall. And for hydrophobic walls, this attraction is greatly enhanced, but also for slightly hydrophilic materials. Okay, now we have a crucial advantage in MD, which we can tune the interaction potential at will. So we generated a purely repulsive gas wall interaction. You see here in the black symbols again. And what this shows is that the attraction, even though the gas and the wall do not have any attraction, if you put a liquid in between, there is a net attraction. And this depends on the level of hydrophobicity. Okay? And the other experiment we did is now we tune the solubility of the gas in the liquid. And if you make the gas very soluble and you compare it with the attraction of the gas to the wall, you see that in this case, the effective attraction to the wall is decreased. So putting together these two experiments, of course, 4 experiments, we see that this attraction is really solvent mediated. And the reason why the gas is attracted to the wall is that if it has low solubility, it doesn't want to stay in the bulk, and it goes to the interface where it minimally disrupts the water structure. In particular, if you have a hydrophobic wall, the depletion at the wall is greater, and there is more room for these unwanted solutes in the bulk. On the other hand, if you have a soluble gas, then this is happening in the bulk, and its concentration at the interface is decreased as compared to what one would expect in a vacuum. So we use this data to do some calculation of the contact angle nanobubble would have in the case of a hydrophobic wall and in the case of a hydrophilic wall. And we notice 2 very important matches with experiment. The first one is that even in undersaturated conditions, so 0.5 and 0.25, you could have measurable nanobubbles, okay, due to this solvent-mediated attraction to the wall. So undersaturated liquids. And the other thing is that depending on the saturation, of course, also you could have, even at hydrophilic walls, the formation of detectable surface nanobubbles. On the other hand, then this is another explanation of some experiments. If you had aprotic liquid, so something that dissolved much better the gases, in this case, you didn't see, experimentalists didn't see any formation of nanobubbles. So to wrap up this part, nanobubbles seem to be long-lived because of a subtle effect of gas accumulation at a wall. This is due to an interplay of the whole trio, so both gas, wall, and liquid, which gives rise, especially for hydrophobic substrate, to an attractive potential. This leads to local gas supersaturation very close to the wall, which could, however, be 1,000 times more concentrated than in the bulk. This provides the necessary attraction to explain nanobubble stability. What I want to say is, since we're talking about confinement here, that here confinement is provided by the liquid-solid interface. So as you could see from the graphs, it's really a nanoscale confinement because it's within the first layer from the wall. So this leads to the second course of our menu today, which is now studying drying induced by a hydrophobic gas in a nanopore. Okay, so far we have seen what is the effect of hydrophobic confinement on gas accumulation, and now we turn the attention into the phase behavior of the water in confinement in the presence of a gas. Why is this relevant for ion channels, which are, so to say, our first preoccupation? Well, first of all, because there is a class of general anesthetics, as has been proposed in this reference, which has rather unspecific interaction with many ion channels. Some of them are even noble gases, so they're not expected to have any interaction, any biological interaction, or very weak one. And the other thing that is interesting in this empirical correlation, which is called Mayer-Overton correlation, is that the anesthetic potency of these gases is correlated to their hydrophobicity. So the more hydrophobic, the more powerful the anesthetic. And so our question is, can these be at least partially explained by the role in drying of nanopores? And so we did it with, so to say, a physical approach, and we did a sort of spherical curve of the ion channels, which is a cylindrical pore in which we can characterize very well the geometry, the size, and especially important here is the hydrophobicity of the nanopore. Always a very standard water model, and a hydrophobic pore. The hydrophobicity has been characterized in detail with these, the measurement of the contact angle. The diameter has been chosen as 1.4 nanometer, which is, so to say, the maximum diameter that is reasonable to think for a biological ion channels. We have considered a very dilute solution in which there's a single hydrophobic particle running around our system, wandering about. Okay, in this case, we have taken parameters for argon. So I will most of the times call it a gas, but it's really one hydrophobic particle. Okay, what happens? Here you have inverted colors, so the argon is in blue. You see that first thing that happens is that the hydrophobic particle is attracted irreversibly within the channel. Here it resides preferentially at the wall, where it induces, you see, low density fluctuations at the wall until, wait a second, and you will see, a sufficiently large one occurs, And this large fluctuation triggers drying of the pore. Okay, so this is the story that these free unrestrained simulation tell us. So let's try to analyze them a little bit better. And here you see the density of water in red in the case without a gas, and in black in the case in which you have a gas. And you see that the first layer, so the peak in the first peak in the density, is locally decreased by the presence of a gas, and that the the probability to find a gas is peaked really at the wall, with the peak, which is even slightly closer to the wall than the first water layer. More importantly, I believe, is the fluctuation. So if you consider the number of water molecules within the channel, well, first of all, they decrease a little bit, but more importantly, the tails corresponding to low density fluctuations of water, so depletion of the channel filling, are greatly enhanced by the presence of a gas. Okay, so these two ingredients together, the location, preferential location of the gas, together with enhanced density fluctuation, facilitate the formation of a critical bubble, which even without a gas is known to form at the hydrophobic wall, because there the energy cost is minimal. So you have really the contribution in the place you need with enhanced fluctuations. So now let's put it into more quantitative form to see even how the kinetics and the thermodynamics changes of the system. In order to do that, we did free energy calculation in two variables. One is the axial coordinate of the gas, which accounts for the penetration of the gas into the ion channel or the nanopore, if you wish. And the second one, which counts the number of waters inside the nanopore. So it accounts for the wetting state of the pore, dry or wet. And so this is the results. So this is a free energy map for our system as a function of the filling and the position of the gas. So you see that this landscape is made of two parallel valleys, one corresponding to the wet pore and one corresponding to the empty pore. And these two valleys have really two plateaus, one corresponding to the gas outside the channel, and the second, which is the lower plateau, corresponding to the gas in the channel. The global minimum is D, which corresponds to the dry channel with the gas in it. And so we imagine a thought experiment in which we start with our system with, we introduce the gas somewhere far away from the pore, which would be A. The pore is initially wet, and we ask ourselves, how do you go from A to D? Okay? And the answer is, there's not a single way, there are at least two ways to go. And the first one is, you first de-wet the channel and then the hydrophobic particle enters, and the other one is the particle enters and then you de-wet the pore. Okay, one can compute the free energy along these two these two paths which locally maximize probability, the likelihood of the transition. And in this case, you see that if you have to de-wet before, you have to overcome a drying barrier. So the formation of the initial nucleation bubble costs some energy. And this energy is, in this case, 2.5 kBt. In the other case, instead, the fact that the gas is inside of the pore and is enhancing fluctuation close to the wall completely obliterates the free energy barrier. So in this case, the free energy barrier is 0.5 kBt. So, yeah. So the net effect of the gas seems to be to bypass the cost of forming the initial bubble, and thus facilitates the drying, the emptying of the pore. Another thing which is important to remark is that without a gas, you would have only the path from A to B, so this vertical path. The presence of a gas, instead, further lowers the free energy from B to D. So this is, so to say, a quantification of the attraction of the gas into the pore. So we thought, well, we need to check whether this mechanism is general or depends on the specific chemistry of the channel. So in this case, we test a neutral pore, so with contact angle 90, and we computed the free energy along the same two paths as shown above, okay, ACD and ABC. And in this case, the picture, the qualitative picture was really the same. So in the case you have first to the wet the channel, there's a much larger free energy barrier, which in this case is of the order of 10 kPt, whereas in the case in which the gas enters first the channel, this free energy barrier is decreased. So you see that the decrease is of 4 kBt, which means that the kinetics is accelerated by exponential of 4, which is, of course, even more remarkable than in the case of a hydrophobic channel. Okay, so the partial conclusion of this second part of the talk is that hydrophobic gases favor drying. As we have seen for the nanobubbles, the presence of a wall already attracts the gas to the wall. If you have a curved wall, this attraction is further enhanced. So gas is hydrophobically attracted into the pore. There, it resides at the wall where it enhances fluctuation. Overall, the effect is to accelerate the formation of bubbles and drying. It stabilizes the gated state. So this might be responsible for the fact that ion channel gating, when it is hydrophobic, is further enhanced by the presence of even dilute solution of hydrophobic gases. And this could go in the direction of explaining the general anesthetic action of unspecific drugs. So overall, the talk of today had a focus, so drying in extreme confinement. And we have learned through simulation and some theory that drying can be induced by nanoscale confinement, and in particular it depends on the size, the geometry and the hydrophobicity of confinement. If you put a third phase, in particular gas, this is crucial in catalyzing drying of nanopores and also in the formation of surface nanobubbles. With that, I would like to thank the group in Rome. Here I should have added Gonzalo, but as you know, Gonzalo is not in Rome, so he could not be in this picture, which is taken close to our department. But I'm very happy that he's now joining the efforts of our group. And I would like to thank you for your attention.
Okay, thanks very much, Alberto. Seminar is now open for discussion. Who has a question? So questions, if you have questions, comments, just raise the little blue hand. If you don't know how to raise the little blue hand, just shout your question.
I have one question, Alberto. Thank you for this nice talk. So when you are speaking about this result, you always concentrate on a water model, right?
Yeah.
How universal are these results? Could you observe them for a simplest possible, let's say, square well, or just Lennard-Jones fluid, and then relate the question: if you have a water, you have also ions. Are there some effect of ions foreseen?
Okay, so actually, this case of highly soluble gases was in a Lennard-Jones. So this effect is rather general, and it depends on the competition. So I think we always use water because people, it makes easier to, so to say, justify a connection with experiments, and with biology in particular, because they don't like Lennard-Jones. But so to say, we have a repeated similar simulation with Lennard-Jones, and the key effect is indeed solvophobicity, or in the case of three phases, the competition between these three effects. So solubility of the gaseous phase and hydrophobicity of the wall. So indeed, this is a completely general mechanism. I think you had a second question, but I probably forgot it.
Once we have water, there are effects of pH, of ions, concentration, electrostatics. How electrostatics is important there? Or
okay, so ions, we're just starting to consider solutions with ions, and so if you mean translocation of ions, what you see, so what we have first seen is that the ions, but this was known before us, ions do not want to pass through a gaseous phase. So they really want to stay in dehydrated states. And so when you consider the free energy barriers of ions translocated through the pore and they're stripped of their hydration shell, then the free energy blows up. So there's a very large barrier. Instead, if you have even these minimal hydration, then the free energy barrier is much lower. Interestingly, some ion channels have a selectivity filter, which is the upper part of the ion channels, which mimics the hydration shell they would have in water. So they have a charge distribution which mimics the hydrogen bond network surrounding the ion. So they select a specific ion because they have a specific structure mimicking the hydration shell of that ion.
thank you. Are there any questions from audience?
Yes, I think there's a question from Nuno. Nuno?
Yeah, okay. So thank you very much for your talk. I was wondering about the role of the length of the channel. And the second question is somehow related. It is, when we are looking to such small systems, wouldn't you expect that the heterogeneities that might emerge, they play a role? So the variance that results, or the variability that results from such heterogeneities will become much more important than when you get in a very symmetric system as the one you just show us?
Thank you very much for the question. So I think the second one is really related to the length. So let me show you a little bit what I would expect here. So the characteristic size that matters is really of the order of magnitude of the diameter. So if you make the channel longer, this doesn't alter the first drying barrier, because the critical bubble that forms at a wall, and it does not fill the length of the channel, but it only fills the other side of the wall. So basically what I'm trying to say is that the size of the critical bubble is determined by the diameter. So as long as you have a channel which is longer than it is thick, then you're fine. So the physics doesn't really change. If you make the channel longer, this branch will become longer. But since that part of de-wetting is simply two menisci moving away from each other, nothing spectacular happens. So this barrier is really determined by the diameter rather than the length. Concerning the heterogeneity, so that's why we limited the length of the channel. Concerning the heterogeneity, this is a very important point. So in biology, there's a strong heterogeneity. But what seems rather conserved, and that's why what I told you just now seems to be relevant, is that in many channels there is a hydrophobic cavity, which is more or less the height of the diameter or something like that. So what I expect is that the function of the channel differs depending on its height. For instance, on the top part, you usually have a selectivity filter, which select the kind of ions. But at least in those channels which exhibit hydrophobic gating, I expect that the hydrophobic cavity really behaves as our channel, because what really matters is not the extent of the bubble, but the fact that the bubble forms. And then there's something else happening in the lower part of the pore. But so this heterogeneity seems to add some function to ion channels. But the fact that a bubble forms anyway determines the blockage of ionic currents. So to say, details change and the function of the channel change, but as long as there is a sufficiently tall hydrophobic part, then I expect the same mechanism to hold.
Thank you.
Okay, so any more questions, comments? Anyone?
I can ask one more question if Paolo allows me.
Yeah, that's okay, Miko.
So when you simulate gas in the nanopore, is it correct that you just had one argon atom? How does it change with the concentration of gas?
Yeah, that's correct, Miko. We're trying to. Okay, so we're in the dilute regime, because typically what happens in anesthesia is that you want a small concentration of a given drug to induce their effect. So you don't want to have, for instance, pressures in volatile anesthetics which are larger than the atmospheric pressure. But indeed, you're right that there can be cooperative effects in that more than one hydrophobic molecule accumulate inside of the pore. So we're still exploring that. There's, however, an indirect effect of the concentration of the gas on these kind of mechanisms, because this concentration sets the typical diffusion time of this atom from the bulk to the pore. Okay, so you can see the kinetics of this dewetting process as a two-step one. Diffusion to the target and overcoming of the barrier. So even in the dilute regime we are studying, you can decouple the effect of concentration because it rules the diffusion to the nanopore, and then the effect of a single atom, of a single particle, hydrophobic particle, into the dewetting. What really surprised us is that a single very small hydrophobic particle is already sufficient to cancel out the drying free energy barrier. So that's why we didn't go immediately into multiple particles. But now we're trying to do that, of course.
Okay, thank you.
Okay, any more questions or comments? Apparently not. So if there are no more questions or comments, let us thank Alberto again. Alberto, thanks very much for a fascinating talk. And we'll be back next week. Miko, who's on next week? Is it Gleb?
Thank you, all of you.
Thank you, Alberto. Yes,
Thank you,
Paolo.
Alberto.
Okay, thanks very much. We'll be back in a week's time. So see you then.
Okay, thank you. Bye-bye.
Bye.
Bye, everyone.
All right.